Alaikum, bete. So let's start doing the continue with the same biological molecules, and we were doing the past paper questions from the year February, March, and May, June 2023. So let's start doing the MCQs from the same year. Let me share the screen with you. I hope you can see the screen. These are all the questions from February, March, and May, June 2023. And we have already discussed a few questions from the same worksheet. So let's finish with the worksheet and let's finish with all the questions from this year. We discussed till this question number 14. So let's continue with the next question, that is question number 15. So, which molecule contain at least 4 double bonds? 4 double bonds. If we talk about hemoglobin and collagen, these are both proteins. And if we talk about just a dipeptide having 2 amino acids, this is the R chain, this is the amine group, this is double bond O carboxylic head, this is 1 amino acid, and the other amino acid, that is again double bond O, OH, N, H, and H. So what happens that for the formation of the dipeptide and for the formation of peptide bond, this 2 hydrogens and oxygen are released out as water molecule. Condensation reaction takes place, and bond is formed between the 2 amino acids, and as a result a dipeptide is formed. A dipeptide is formed. How is it formed? Between the 2 amino acids, C, N, H, H, R, H, C, double bond O, because hydroxyl is removed, N, H, because amine group's 1 hydrogen is also removed and peptide bond is formed over here, and C, R, H, C, double bond O, OH. So this is a dipeptide, and a peptide bond is formed between the 2 amino acids, and this is the peptide bond, and that's a dipeptide. So collagen and hemoglobin are made up of so many amino acids. They are quaternary structures. They have this has collagen molecule has 3 polypeptides. Hemoglobin has 4 polypeptides, and they are made up of so many amino acids. If they are made up of so many amino acids, and we have just drawn 2, and in these 2 amino acids, when they are bonded, there are 2 double bonds, 1 and 2. So the second double bond. So if within 2 amino acids there are 2 double bonds, then what about these hemoglobin and collagen made up of so many amino acids? They would be having a lot of double bonds. So they both are correct. If we talk about unsaturated triglyceride, and unsaturated triglyceride can be a monounsaturated triglyceride. So if we draw the structure of a triglyceride, this is glycerol, having 3 hydroxyls, and this is a triglyceride having 3 fatty acid chains, and the long hydrocarbon tail. And they are seeing at least 4 double bonds. So this, from here hydrogen and hydroxyl are removed. Condensation reaction takes place. 3 water molecules are produced this way. And this is what a triglyceride is. If it's a triglyceride, unsaturated triglyceride, and if it's, let's suppose it's a monounsaturated, there is only one double bond in one of the fatty acid tail. Then one double bond is here in the carboxylic head, other is here in the next carboxylic head, third is in the third carboxylic head, and fourth is because of the unsaturation. Anyone, it can be more than one double bonds, but if we consider the least, then there will be at least one double bond in the fatty acid tail. So there are, it is correct then that it can also have 3 double bonds, 4 double bonds, at least 4. It has 1, 2, 3 already in its carboxylic head, and the fourth one would be in the unsaturated fatty acid tail. So all of these 3 have 4 double bonds. They would be having more than 4, and it would be having at least 4 would be must present in it, in unsaturated triglyceride. Next is the diagram shows the amino acid glutamic acid, because it's glutamic acid, because its R chain is, this is the R chain. This is the generalized structure of the amino acid: carboxylic head, amine group, hydrogen always attached to one of the carbon, and that's the different one. So this is the R group, and that's the question is, so it would be D. The diagram shows 3 interactions that hold protein molecule in shape. The 3 interactions, we can identify them. This one, the dotted line between partial positive and partial negative, this is hydrogen bond. This is the disulfide linkage, clearly visible. The 2 sulfurs are connected. And this is ionic bond between one positive and the other negative ion. So they are asking which row identify these interactions. So the question we have already solved. Let's see. Interaction 1, hydrogen bond. Interaction 2, disulfide linkage. Interaction 3, no, ionic bond. Okay, this is B, because disulfide is also a type of covalent bond. So hydrogen bond, covalent bond, and ionic bond would be the correct options. So disulfide is also ionic bond, covalent bond. So hydrogen bond, covalent bond, type of covalent bond, that's disulfide linkage, and ionic bond. B is the correct option. To estimate the concentration of glucose in an unknown solution, equal volume of a range of known concentration of glucose were each mixed with the same excess volume of Benedict solution. After mixing, the solution were placed in thermostatically controlled water bath at 90 degrees Celsius for 3 minutes. The unknown solution was then treated in the same way, and the colors of the known and unknown solutions compared. What is the independent variable in this procedure? Independent variable, temperature we have already decided 90, so that's not independent. And the color change does not depend upon the temperature, because the temperature for Benedict is always 90. Concentration of glucose is the independent variable, because the color is dependent on the concentration of glucose. More the concentration of glucose, more will be the color of the red brick precipitates, or bright brick, or bright red precipitates, or if it is not even red, orange or yellow, yellowish orange. So the strength of the color and the different ranges of color is because of the concentration of glucose. So color is independent over here, and concentration of glucose is independent variable. Final color of solution, temperature is fixed, that's 90, it's not independent. volume of glucose, we fix it always, it is same. Volume of glucose solution, it's also fixed. Usually we use 2 centimeter cube for doing it in the laboratory. Final color of solution is also not independent because it depends on the concentration of glucose. So all these three are incorrect. Only the concentration of glucose is independent. Which statements are correct for amylose and also for amylopectin? You know, they both are made up of alpha glucose. They both are made up of. They both have 1,4 glycosidic linkage, and this is amylose is linear, but this is branched. That's the difference. And this is branch. So if we talk about correct for both, so let's see, they're carbohydrate molecules, correct. They are formed by condensation reactions, correct. Every time condensation reaction takes place and water is produced, so that is also correct. They are linear molecule, that is incorrect. I've told you that this is branched. It has branches, 6 branches. It does not have branches. This is only linear. So amylose is linear, not amylopectin, that is incorrect. They contain alpha 1,4 glycosidic bonds, that is correct. Alpha and 1,4, that is also correct. So 1, 2, 4. Now, which statement is correct comparison between saturated triglyceride having no double bonds and unsaturated triglyceride molecules of approximately the same molecular mass? So unsaturated is with double bond, and you know, whenever double bond is formed, the number of hydrogen atoms get reduced. Like if this is a hydrocarbon tail, or it's another hydrocarbon tail, there's a double bond in it, it would be having hydrogen, but over here only one hydrogen will be present. Again, here only one hydrogen will be present. Here would be two, because carbon can be bonded to 4. So the number of, in case of double bond, the number of hydrogen, 2 of the hydrogens are reduced. The number is reduced to 2. So keeping this in mind, that's saturated and that is unsaturated. Keeping this in mind, this is unsaturated. Let me draw it with another pen so that you will understand it. This is unsaturated. So you can see 2 of the hydrogen are reduced. There are less hydrogens in case of unsaturated as compared to saturated, because carbon is already double bonded. So they are saying unsaturated triglycerides have more double bonds and have more double bonds and fewer hydrogen atoms. Then that is absolutely correct. Let's see the rest. Unsaturated have fewer double bonds? No. Why would it be then unsaturated? It would be having fewer double bonds because saturated does not have. Incorrect. Unsaturated triglycerides have more double bonds and more hydrogen and no, not more, less. Unsaturated have fewer double bonds. There's no chance of fewer double bonds because it is called as unsaturated because it has the double bonds, and saturated is not called as saturated because it does not have. There's no comparison in double bond. It does not have double bonds and it has double bonds. So there's no comparison of more and fewer in case of double bonds, because the one which is unsaturated has double bonds and the one which is unsaturated has double bonds, and the one which is saturated does not have double bonds. So there's no comparison in their number of double bonds. A is the correct answer. The diagram represents a molecule from a surface membrane. That's a phospholipid, phosphate head, glycerol, and these are the Fatty acid at the hydrophilic end of the molecule, incorrect. That's at the hydrophobic end of the description of A, fatty acid at the hydrophilic end, that is wrong because fatty acid is at the hydrophobic end of the molecule. Next is hydrophobic end of the triglyceride molecule, B, that is correct, the hydrophobic portion, that is correct. Even then, let's see, hydrophobic end of triglyceride. Oh, this is not a triglyceride molecule. This is a phospholipid. So this is also incorrect. This is also incorrect. See, they confuse you this way. Hydrophobic end, yes, it is, but it's a phospholipid molecule, not a triglyceride. That is incorrect. This is hydrophobic. Then hydrophobic end of the glycerol molecule. This C is the hydrophobic end. No, it's the hydrophilic end because it has phosphate attached to it. So it's the hydrophilic. That is wrong. And D is the phosphate group at the hydrophilic end of the molecule. That is correct. This is the hydrophilic portion and where phosphate is attached. Which row correctly shows level of protein structure that can be held together by each level of protein structure that can be held together by each type of interaction? So hydrogen bonds are present in primary, secondary and tertiary, not in primary. Incorrect. Let's move forward. It is present in secondary, hydrophobic interactions, not in primary. Primary has only peptide bonds, so that's incorrect. Hydrogen is in secondary, tertiary, correct. Hydrophobic in tertiary, and covalent bonds which can be held together by each type of interaction. So primary has peptide and tertiary has disulfide linkage, that is correct. Covalent bond which are peptide in the primary and disulfide in the tertiary. So that is correct in all aspects. Hydrogen bond, secondary, tertiary, correct. Hydrophobic, tertiary, correct. Covalent bond, primary, and no, they don't hold the secondary structure. They hold the primary structure. Secondary is held by the hydrogen bonds. So that is also incorrect. Which molecule contains at least 3 double bonds? Again, the same type of question. This would be having, this would be having. But if we talk about saturated triglyceride, a saturated triglyceride would be having, again, 3 double bonds like with the one which we discussed a few minutes ago. We discussed it over here. I do the, yes. If it is a triglyceride and saturated one, then there will be no double bond in the fatty acid tails. So there will be only these 3 double bonds, 1, 2, 3 in the carboxylic head. So at least 3 when they are asking, so it means that triglyceride which is saturated also fulfills the demand of 3 double bonds. So all these have, they have for sure more than 3 double bonds, but this also has 3 double bonds, so D. And the last MCQ from February, March and May, June 2023, it says that which diagram correctly shows hydrogen bonding between 2 water molecules? Now, hydrogen bonding between 2 water molecules, that's this sign is correct. They are not fully positive negative, they are partially. So these two are incorrect because that should always be this way, because it shows, this shows partial positivity and partial negativity. They are not fully charged this way because bond is being formed, but the charge is because of unequal sharing of the electrons. So that's partial. Why hydrogen is partial negative? That is also wrong. It's absolutely this one. Partial positive, positive, negative, positive. Hydrogen bond is being formed, and this is oxygen and hydrogen. And this is how you can see that between 2 water molecules, there was an MCQ like this. Between 2 water molecules, there can be maximum 1 hydrogen bond which is being formed. Okay, or maximum 1 hydrogen bond is formed. If this oxygen is going to make bond with the hydrogen, it is not going to make bond with this hydrogen, it is going to make bond with hydrogen of another water molecule. So maximum, because the configuration is in this way now that these cannot make another hydrogen bond. They can with the same molecule, they can make with some other molecule of water. So these are all the MCQs of the year 2023, May, June, and February, March. Soon I'll be discussing the MCQs of October, November, after October, November, May, June 2023. Inshallah. These were the questions of May, June, and February, March, and as I've just told you that I'll be discussing the MCQs from October, November from the same topic, biological molecule, after October, November session, inshallah. I hope everything is clear to you. If you did not get anything, you can ask me in the comment section, and you can like my videos, and you can share my videos, and you can watch, and you can ask questions. Thank you very much. Allah Hafiz. See you with the next video. Allah Hafiz.